There's a lot of time during the meeting to ask more questions, and Dr. Glut will be around. All right, now I'm not Paula Bontempi, but I'm the ASLO co-chair, and it is a pleasure for me to introduce the G. Evelyn Hutchinson Award, which has been presented annually since 1982 to recognize excellence in any aspects of limnology and oceanography. This ASLO Award is intended to symbolize the quality and innovations towards which society strives, and to remind its members of its goals. In lending his name to this award, Hutchinson asked that recipients be scientists who made considerable contributions to knowledge and whose future work promise a continuing legacy of scientific excellence. If you check out the ASLO website, you will see that it is a fine group of scientists who has received this award. Dr. Liz Harvey will present the award on behalf of ASLO. Dr. Harvey got her doctorate degree at the University of Rhode Island and is currently a professor at the University of New Hampshire. Please join me in welcoming Dr. Harvey.
Hi, everybody. First, I want to thank you all for being in attendance today. It's not every day that I get to present an award to someone whose work and collegiality I admire, so I really appreciate this minute of your time. This year's Hutchinson Award winner, Dr. Elizabeth Kujawinski, has transformed the way that ocean scientists observe the exchange of dissolved chemical compounds that mediate ocean processes. In particular, Dr. Kujawinski has been a pioneer of the development of methods and computational tools that use data from ultra-high resolution mass spectroscopy to characterize dissolved chemical landscapes of the ocean and investigates the implications of these compounds to biogeochemical processes. Throughout her career, she has amassed an impressive publication record, including work examining the impacts of chemical dispersants used in the Deepwater Horizon oil spill, looking at metabolite exchange between phytoplankton and heterotrophic bacteria, and the cycling of important organic sulfur compounds. In addition to her research achievements, Dr. Kujawinski is an active leader in the marine science community, serving on a variety of local, national, and international committees, leading numerous expeditions as a chief scientist, and developing programs that foster diverse entryways into a scientific career. As a senior scientist at the Woods Hole Oceanographic Institution and the current director of the Center for Chemical Currencies of a Microbial Planet, an NSF-funded science and technology center, Dr. Kujawinski is well positioned to continue to succeed at the highest level and further evolve our understanding of how dissolved metabolites mediate marine ecosystem function. And it's now my pleasure to congratulate and present the 2024 Hutchinson Award to Liz Kujawinski. Thank you.
Okay. I'll put it down. Okay. Okay. Thanks, everybody. Really appreciate you all being here. I'm incredibly humbled to be giving this award lecture, and it's really an opportunity to talk about the work that my group has been doing and to celebrate that with all of you today. I thought I would start by telling you a little bit about G. Evelyn Hutchinson, the person for whom this award is named. He was a British ecologist. He was a professor of zoology at Yale University for many, many years. He's considered the first to treat ecology as a quantitative science, combining math, experiments, and observations to try and understand ecosystems. Interestingly, he anticipated many of the impacts of climate change as he thought about what a changing carbon dioxide concentration would look like in the atmosphere and the impact of that on biological systems. And three words that come out over and over as you read tributes to Dr. Hutchinson and as you read about the many tributes that have been given to him over the years are three important words that I think all of us can really rely on and connect with. He was collaborative, he was generous, and he was unafraid. He mentored over 30 PhD students. He was generous with his time and his ideas, and he was unafraid to go forward and do new things and explore new territories. As I reflected on how I would like those three words to describe the work that I do, I was struck by the fact that there are two people. There are two people whose loss looms large for some of us at this meeting this year. The first is my former graduate student, Dr. Maya Bhatia, who is a glaciologist at the University of Alberta who was lost in field work this past August. Her contributions to glaciology, community-driven science, mentoring, and science are so impressive and will be sorely missed. Dr. Jan Amend was an external advisory board member to our STC Center, and in all of my interactions with him, he was generous, collaborative, unafraid, and incredibly supportive of our efforts. One of my friends pointed out to me that everyone has a Jan story, and similarly, everyone has a Maya story. So if I could take just a moment to wish those safe seas and give ourselves a minute to remember that life is short. And so wherever you can in your science, please be collaborative, be generous, and be unafraid. Okay, the rest of my talk will be just hopefully a brief chronology of our science journey, pointing out some areas in which we've had to take some leaps, where we've tried to learn some new things, where we've worked with lots of exciting people and done some fun stuff. So I first went to college to major in chemistry because I thought it would be fun to work with molecules and solutions that were colored. I went into organic chemistry, for which nothing is colored. I then went to graduate school and worked under Jim Moffitt and John Farrington, thinking about the fate of polychlorinated biphenyls in New Bedford Harbor. I wanted to save the world, as so many of us do at that time. When I became a postdoc with Mike Freitas and Pat Hatcher at Ohio State University, I was starting to learn new tools about how to characterize organic matter, and I was really excited about this connection between pollutants and organic matter at the molecular level. My first job was at Barnard College in New York City, working at an all-women's school and working with undergraduates to try and apply some of these tools to understand natural organic matter cycling. At that point, I got married, and it turned out that sharing the same zip code, if not the same house, was desirable for long-term marital happiness, and so I moved to the Woods Hole Oceanographic Institution. And I've been there ever since, and it's been just a fabulous ride. This is my view of what I was trying to do at that time. I was thinking about the global carbon cycle. I was really excited about dissolved organic carbon. I wanted to do something that helped me understand the cycling of that system relative to global carbon balance. And so we began to work on different projects in that vein. When I came to Woods Hole, one of the things that I had been working on since my postdoc and then into my first job was trying to understand the composition of organic matter. And we had pioneered a tool, ultra-high resolution mass spectrometry, that uses this long acronym, the Fourier transform ion cyclotron resonance mass spectrometer. And we were getting unprecedented views of natural organic matter composition. So at this kind of resolving power, we were getting on order 5,000 peaks resolved to baseline resolution. This was really exciting and heady stuff at the time. And what you can do with that kind of mass accuracy and precision is to assign elemental formulas really, really precisely. And so you can use carbon, hydrogen, oxygen, nitrogen, and sulfur, and you can plot them on what my husband calls the magic dot plots, where you have oxygen to carbon on the x-axis and hydrogen to carbon on the y-axis. And different types of molecules have characteristic, falling characteristic portions of this plot. And so you can tell the difference between, say, lipids and lignin and carbohydrates and so on. And so our goal was to then begin to use this kind of technology to begin to understand what was happening. So this begins phase one, the FT years. And enter the two folks who have really been the cornerstone and platform of my entire program at Woods Hole, Krista Longnecker and Melissa Keto-Sewell. And we were fortunate enough to get an MRI proposal funded by NSF, and it was, allowed us to purchase our first FT ourselves. And so this is a picture of the FT. We specifically don't have on the side panels because, as we decided, we didn't want the black box approach, so we could see all the innards. And our first years were dedicated to trying to get good data. How could we be reproducible? How could we get reliable results? How could we interpret that data? This was sort of a new data stream, and we wanted to understand how to do that. And then, could we use that kind of data to answer our questions about organic matter, the environment, processes, and so on? So this became my view of the aquatic carbon cycle. Notice the use of Comic Sans, a font that has sadly gone out of favor over the years. And you can see that really what I was thinking about at the time was the separation between humics and biological material. And then we're not too sophisticated in the biology here, apologies to my colleagues in that space, but we were trying to think about these types of pools of material occurring or interacting with one another and with different organisms on a molecular level. So we were trying to get to chemical reactions, products, and reactants. Okay. So our first approach was instrument optimization and data analysis, and you can see here the one and only marital paper here, Kujawinski and Bain. But in addition, we were trying to understand how we could reproduce our data, how we could analyze the data, and then we started to apply it to different environments. So the paper here on your left is looking at the coastal versus the open ocean. The paper on the right is one of Maya's papers from graduate school looking at organic matter associated with ice sheets. We also were thinking about it thinking about DOM occurring as a function of different microbial communities. And so I'm just going to show you a very little bit of data here. These are the sort of haystack plots that are very common with FT data. On the top is our initial condition, our dissolved organic matter collected from the T0. When we add bacteria, protists, and a substrate, we get the second one, which doesn't look very different than the first one. And then if you omit the protists, the unsung heroes, in my opinion, of the microbial world, if you just have the bacteria and the substrate, you get something that looks a little bit different. I will also note for you the year in which this was published, because I am first author, and it takes forever for me to get anything out the door. Okay. So we took the same Van Krevelen plot that I showed you before, and we plotted all of the points. And then in black, I'm showing you the features, so to speak, that were unique to the bacteria only. And in red, I'm showing you the features that are unique to the whole water. And we start to try to understand what these molecules are. And you can see that we can't be very precise. We can get a sense, well, maybe the bacteria are making some lipid stuff. Well, so the stuff that's whole water indicator really looks like the background material, so maybe that's not really telling us much about that biology. But this is where we were. And then two big things happened in the group. The first one was that I went to a metabolomics society. And I point this out for the younger folks in the crowd. Don't be afraid to go to conferences that are outside your comfort zone, because they can really transform the way you think about your own science, as well as your place in the broader scientific endeavor. And when I went to the Metabolomics Society meeting in 2008 in Boston, I realized that folks there were using the same kinds of data that we were generating for the ocean. They were using that kind of data to understand disease, progression of disease, biomarkers of disease, and so on. And I started to think about how powerful that kind of an approach could be as we thought about changes in the ocean. In 2010 was the Deepwater Horizon oil spill, which many people in oceanography became involved in. And let me focus on that for just a moment. So here on the left-hand side is a schematic of the Deepwater Horizon spill, where at the bottom you have the damaged wellhead, at the top you have the at the top you have the boats and the planes in their response, and in between you have the plume of oil coming up, and then there's sort of a left-hand turn, and that's the deep water plume. And this is work that was led by Melissa in my lab, but also our summer student fellow, Angie, who is now faculty member at Pacific Lutheran University. And what we saw using our typical data sets was that if you looked at the seawater DOM above, in an area that was not oil impacted, it looked exactly like the background. But if you went to this plume that was turning off to the left, there was this very dominant molecule, or this one dominant feature that showed up with the right molecular weight for the one of the dispersant components. But it turns out it's not enough to just say that it's there. You have to be able to quantify it, map it, and understand where it is. And so Melissa and Angie turned around and made a quantitative method which allowed us to really focus in on where the dispersant was in the immediate aftermath of the spill, and then in that paper we're able to track it over time and show that it dispersed throughout the Gulf of Mexico. So where we were at this point was we realized we wanted to do something that was more connected to systems biology, because we wanted to be able to sort of diagnose the biological system of the ocean. And we needed to be quantitative, because it wasn't going to be enough to just say what was there versus what wasn't. So we began a pivot to metabolomics, and that forced us to start thinking about compound identification and quantification. We started thinking about the difference between dissolved and particulate. We started thinking about sampling strategies, new methods, new optimizations. This is, again, led by Krista and Melissa, and now we enter our other long-term technical staff member, Ms. Gretchen Swar. Two additional things happened that were important. First, NSF was generous to give us another MRI proposal to purchase a quantitative mass spectrometer, and that's the picture that you see there. And the second was that we got a grant funded from the Moore Foundation, who gave us enough money to buy standards. And that really, I really can't overemphasize how important it was to have a pot of money to buy all those crazy standards that are $10,000 per milligram, right? So there was just no way to do this any other way. Okay, at the same time I was sort of writing this review paper in the background, and I tell this story about how there were two piles of papers on my desk, one written by chemists and one pile written by biologists, and I wanted to do something that was in the middle. However, full disclosure, I am a chemist. I don't really understand biology. I do the very best I can. But in order to make this work, in order to be able to move this forward, collaboration was going to be required. This is the method that we started off with, where you basically take seawater, you remove the particles, you acidify to make it more amenable to solid phase extraction, you elute with methanol, and you run it on liquid chromatography to spread everything out, and mass spectrometry to get those individual peaks. This is a method that was pioneered by Thorsten Dittmar, and then we worked on it in terms of its application towards metabolite analysis. There are two different ways you can do metabolomics. You can do targeted metabolomics, where you quantify with external standards, or you can do untargeted metabolomics, where it's more similar to those haystack plots I showed you, where you kind of hope for the best and you see everything that the mass spec can show you. Just to focus this presentation, I'm going to be in the targeted section only for the rest of the talk, but I'm happy at any point to talk about some of the cool work that folks in my lab, like Aaron McParland and others, have done using untargeted metabolomics. Okay. All right, I'm going to use this background. This is the map of all of KEGG, the biochemical pathways in KEGG, and they're colored by different pathway types. I'm going to use this as a backdrop to orient us according to the molecules that we now measure relatively routinely in our lab. This is a big figure, so I apologize for the time it takes. Okay, these are the different pathways to orient you a little bit. I can't keep this in my head, so just in case you can't either, here they are. Okay, in the method that I just showed you, the molecules that we can measure is on order 35 to 40, and they're in all the black circles. So you can see that the molecules are scattered across different biochemical pathways, and we can get lots of interesting ideas and thoughts about what's happening by comparing and contrasting our thoughts. So we decided to take this method out on the road, and I'm going to show you two examples. The first one is we were thinking about spatial gradients. So there's two cruises here that were both conducted, or both, the data was worked up by my former graduate student, Wynne Johnson, who's shown here. And what I'm going to try and show you here in this one data plot is the comparison of two metabolites, one dissolved riboflavin, which is vitamin B2, and the other one is dissolved MTA, or 5'-methylthioadenosine, which is the entrance to methionine salvage. Relative to chlorophyll in the upper left, you can see that these two molecules are following chlorophyll sort of generally, particularly riboflavin. And dissolved MTA is really interesting because we see it very nicely in the Atlantic, but not in the Pacific. So that's going to be kind of interesting. We can also look at particulate MTA, and MTA is an important molecule in cells because it is important for helping to regulate methylation of proteins in DNA, and you can see that it follows a different track. And so this also started to give us a clue that we were going to need to understand the dissolved and the particulate independently as well as together. Okay, we took that southern portion of that cruise. I want to give an example of a collaboration that has been really, really formative in our thinking. This is the Deep Dom cruise. It was the southern portion of what I showed you before. It went from Uruguay to Barbados. And we invited and were joined by a number of people who contributed their expertise and their data for helping us understand how these different aspects were going to fit together. At the same time, we were working on thinking about how we could understand the changes in metabolites in one point in time, one point in space, but over a long period of time. And here we were fortunate to be invited to join Bioscope, which is led by Craig Carlson at Santa Barbara. And the purpose here was for us to put our metabolite results within the seasonal context of DOC that Craig and Dennis had showed nicely over so many years. Our first papers from these are just now starting to come out. This is Krista's paper that came out earlier this month, looking at pantothenic acid, which is vitamin B5, and its seasonal dynamics at bats. And you can see that there is a seasonal accumulation of pantothenic acid that occurs over this 3-year time course. And so we're starting to get some ideas about what's driving some of these different metabolite transitions. Okay. At the same time, because you can never do anything in this job one at a time, we started forming collaborations to begin to think about the sources and sinks of these metabolites, and for this we need to go to laboratory cultures. And so I'm going to show you a couple of papers that have come out looking at this kind of work over the last couple of years. So we first started off thinking about the dissolved material produced by Thalassiocyra pseudonana. That was Krista's work. And then my former postdoc, Cara Fiore, was involved in looking at Micromonas and Synechococcus. At the same time, we also looked at Prochlorococcus across different ecotypes and showed some really interesting differences as well as similarities across those. And then at the same time, we were working with folks who could work on heterotrophic bacteria. So what do these organisms take up? What do they put back into the water after their work? One of the areas in which we're actively working and still have some room to grow are the methods that we use. And I'm going to summarize them for you here. So the one that I've shown you so far and has been the one that generated the data that you've seen till now is the PPL method. It's very good for molecules that adsorb to this resin. They're aromatic or they're kind of biggish. You kind of need them to be about semipolar. If they're too polar, they don't show up. If they're too nonpolar, they don't come off. The sample volume we were looking at was somewhere between 1 and 10 liters. Higher volumes are for oligotrophic regions where you have very little material. We can see reliably about 38 different metabolites. The detection limits are sub-picomolar to low nanomolar. And we've been using this method since about 2013 in the field. When we use this method in cultures to look at biological material, what we get frustrated by is that we can only really account for about 1, maybe sometimes up to 4 or 5% of the material. So that's been a frustration. And so we want to deal with 3 challenges if we're going to design any new methods. 1, we want to get the sample volume down because we can't do space-time resolution very well with this kind of volume. We want to be better at looking at molecules, particularly those that are really interesting to us but don't fall into the zone of good for this method category. And then we really want to get better at accounting for the biological DOC in cultures. Otherwise, this is basically just a boutique method, and really, what are we all doing? So enter 2 additional members of the group, and I'm going to highlight Melissa Sewell here as one of the sort of intellectual leaders of this exercise. Brittany Widner and Katie Halloran are 2 members of my lab. Brittany is a former postdoc, and Katie is a current graduate student. And between the two of them, they have taken on the challenge of modifying some of our methods with one particular commonality, and that is that we now add a benzene ring to different functionalities to make them better adherents to the PPL resin that we use. And so in Brittany's method, we use benzoyl chloride to modify amines and alcohols. And in Katie's method, we use aniline to modify carboxylic acids, some carbonyls, and phosphates. This has a couple of implications. Number one, as I mentioned already, it increases the adsorption to the PPL resin. It also makes our chromatography a lot better. Instead of having everything bunched up right at the front, now it's kind of stretched out along the chromatographic gradient. And then really importantly, it generates an isotopically labeled standard for every single analyte. So anything we want to look at, we can now quantify absolutely. And Brittany's method is published. Katie's method is in progress. She's hoping to graduate at the end of the semester. And then Brianna Garcia and others in the lab are getting ready to publish some general improvements to Brittany's method. Okay. So this is where we were before we designed the new methods, just to reorient you. Oh, geez. Went zipping. And this is where we are now when we add the two new methods. I've taken off the descriptors of the pathways because it got a little bit out of hand and you won't be able to see all the dots. The ones that are in squares are unique to the benzoyl chloride method. The ones that are in triangles are unique to the aniline method, and those that are gray are overlapping. We can see them in more than one method. What I want to highlight for you now is that we have really good coverage of a lot of the pathways of central carbon and nitrogen metabolism, and that is going to be really interesting for us as we move forward and start to understand how these molecules are being exchanged between different organisms. You'll also see that there are some areas in which we don't have coverage. Those are either areas for future collaboration, future method development, and so on. But those are definitely still on our horizon, but they're not going to be part of the next phase. So how do these methods now compare to the original PPL? They address different types of functionalities, right? So now we're looking at different kinds of molecules. One of the things that we're very, very happy about is that we've solved the volume problem. So now our sample sizes are somewhere between 5 and 25 milliliters instead of 1 to 4 liters. Turns out when you work with collaborators, they get really antsy if you take most of the Niskin bottle or the entire culture batch to do one measurement. This works much, much better. We have not sacrificed detection limits. We're still in the 1s to 10s of picomolar, low nanomolar. But what is also really exciting about these two new methods is that when we apply them to culture, biological DOC, we are now accounting for almost 20% of the carbon that's produced. And that's a game changer, right? That tells us that not only are we looking at molecules that are important, we're looking at the ones that are likely produced in the field, and so we can have targets for future analysis. We've applied the benzoyl chloride method in the field and gotten some really nice results. My Bridge to PhD fellow, Herman Garcia, had a nice poster yesterday. Feel free to go see that again if you'd like. We have not yet applied aniline in the field, but we're hoping to do so in the coming year. Okay, so that's where we are. Enter phase 3. We now have good molecules. We have some nice methods we're interested in using. We're doing pretty well in terms of applying things to the field. But this is not going to get us where we need to be. We're not going to be answering the grand challenges put forward by Oscar and Scott earlier today. We're not going to be able to really get at some of these core questions if we can't move beyond the initial pieces. So enter Marianne Moran, what my daughter calls my best friend at work. And we started by co-sponsoring and co-organizing a more foundation-sponsored workshop to look at the intersections between dissolved organic matter analysis and microbiology. From that, we wrote a proposal to CIFAR, the Canadian Institute for the Advancement of Research, to try and establish a working group that would explore labile dissolved organic matter composition. We got to the finals, but we didn't get there. And so a couple weeks after we learned we had not made the finals, we decided to go for an STC. So in that proposal goes to the National Science Foundation, the Science and Technology Center grant, and it's outside core programs, and so it's an opportunity to think more broadly and to move into other areas of resources at the National Science Foundation. And I'm going to tell you a little bit more about C-Comp as we move forward. But I want to acknowledge the contributions and investment of the National Science Foundation for helping us establish the Center for the Chemical Currencies of a Microbial Planet. So it turns out when you're going for a big award like this, you can get graphics help, and so your graphics begin to get a lot nicer. This is where we started our thought process. So we're interested now in focusing on that labile DOC component. It's a very small amount of the standing concentration, but it's about a quarter of the Earth's carbon, it's about a quarter of the fixed carbon on Earth every single year. And it's a group of molecules we know very little about in terms of their identity. We know very little about their dynamics. And all of the rates and dynamics of these molecules are not adequately described in models because the turnover is so fast that it oftentimes is hidden in the time steps that are used. In order to look at that pool, we have taken a very mechanistic and reductionist approach in our first couple years, where we're thinking about all of this happening on the scale of a droplet, so molecules and individual organisms. And the goal then is to take that kind of mechanistic information and translate it into an understanding of the Earth's planet and the carbon cycle. So we're really seeking, as our grand challenge, to understand the network of the surface ocean microbes and molecules sufficiently that we can understand the rules of that network and then constrain its sensitivities to a changing ocean. Did I get, sorry. Yeah, okay. So our research in C-Comp bridges a number of different scales, and I want to here highlight all of the different folks that have joined us, from biologists to chemists to numerical modelers to bioinformaticians, postdocs, bridge to PhD fellows, graduate students, undergraduates, and everyone. And what we do is we explore microbes, whether they're individual cultures or mixed communities. We explore their chemical impact. We try to do it with high throughput and efficiency, and then through modeling, we try and connect it to our understanding of the current and the future environment. A good portion of C-Comp is broadening participation and diversity within the ocean sciences. We feel this is the critical societal problem of our time, and we are not going to get very far if everybody on the globe is not given the opportunity to participate in its solution. And so we take an all-hands-on-deck approach to understanding how we can bring as many people to bear on this question as possible. We love this braided river analogy that was published by Berha in Nature Geoscience a couple years ago because it highlights what's important for us, which is multiple roads of entry into STEM and the ocean sciences. It highlights a grounding in education research, and here I want to highlight Professors Erin Dolan and Laura O'Dwyer, who have joined us in education research, and our education coordinator, Dr. Victoria Centorino, in bringing all of these different programs to fruition and moving them forward in a method that allows us to bring in best practices. I want to highlight our Bridge to PhD fellowship program. The current cohort that is in their second year of this fellowship program is here in the audience presenting posters at Ocean Sciences, and I'm really happy that they were able to join us. So what's coming up for the future? First, I think that our new methods are going to let us make better measurements with the grand goal of getting to rates. So what I'm showing you here is pantothenic acid, which is one of the molecules that's highlighted in Krista's paper, and the difference between the method that we were able to use using the PPL, that's in red, relative to the blue, which is the new benzoyl chloride method, and you can see replication and much better spatial resolution, which is a real success of that method. We can also use it to look at different molecules. For example, taurine is inaccessible to one of our inaccessible to the PPL method, but shows up beautifully with benzoyl chloride. If we can begin to understand these molecules on the appropriate spatial and temporal scales, we can pull out rates. That gets us into turnover times. That gets us into the modeling space and starting to make some really big impacts on understanding. We can also apply these methods to understand what kinds of molecules different organisms are putting out. And this is some early data from the center showing 6 different autotrophic phytoplankton, and they're sort of the top 10 metabolites that they put out into the dissolved phase. You can see that they're all pretty different. And so what we need to do is we need to do this better and do this more systematically so that we end up having the most comprehensive lexicon of these molecules that we can for future understanding of how the microbiome is exchanging substrate, energy, cofactors, and signals. A really critical component to CCOMP, and I think one that is shared by many of you, is the idea that we should be able to move across scales, and in this case using numerical models. We want to be able to move from the smallest spatial and temporal scales all the way up to the basin and global ocean, multi-decadal scales, to be able to take the understanding that we learn from the small scale and use it to understand and predict what's going to happen on the larger scale, and then to go back again. How is the microbiome going to change under these different conditions? Setting up ways to have these different models talk to each other is, according to one of our collaborators, the wild, wild west of numerical modeling. And we're really eager to engage with these communities to help and provide ground truthing data to make this a possibility. And lastly, if we can achieve the potential that all of these methods and these scales bring to us, we can get to the idea of generating figures like this beautiful one from Dennis Hansel's review paper in Annual Reviews, where here he's mapped DOC as a function of all of the ocean basins. What if we could do the same thing with metabolites and microbes? And that's the goal of biogeoscapes and other international initiatives. And I think with the work we're doing through CCOMP, but also through all of the collaborations with all of you all, I think we're about to be ready to do this. What is the state of the ocean? What are the rates at which these important processes are going to change? And what is the fate of carbon under these different circumstances? So here I'll just take a couple minutes to say some thank yous. I want to highlight the financial support that I have received over the years. At the top, the NOAA Omics and the Gulf of Mexico Research Initiative have funded 2 areas of research in my lab that I wasn't able to talk about. One is coral reef science, together with Amy April, another faculty colleague at HUI. And the Gulf of Mexico Research Initiative has funded some work on oil degradation that was done in collaboration with Helen White from Haverford College. I want to thank the Gordon and Betty Moore Foundation and the Simons Foundation for having given me the freedom and the ability to follow dreams and do crazy things and have some support behind that. Throughout all of that time, the National Science Foundation has been an important investor and an important partner, and I want to thank Don Rice, my first program officer, all the way to Hetty Edmonds, our current program officer, for being supportive of not only our work, but that work of ocean sciences in general. And then I want to thank the Woods Hole Oceanographic Institution. It's where I did my PhD, and I left as quickly as I could, and I thought I would never, ever, ever return. I came back in 2004 with my husband, and I haven't left, and I honestly don't really think about leaving very often. It's a really amazing place to do science. It was an amazing place to raise my family, and it's just been, and I want to acknowledge the support of all of the leadership, my current department chair, Ben Van Moy, and then the current leadership of HUI, Rick Murray and Peter Domenical, for making it a great and supportive place to work. I want to close, and I'll leave this slide up here, inspired by the folks who raised me, my parents, Joe and Frank Kujawinski, and the family that I am trying to raise with my husband Mark, my son Alex, and my daughter Claire. And then those of you who know me well know I stress about more than one thing at a time. And one of the things that's really been bothering me and making me nervous about giving this talk is that I desperately want to do justice to everybody who has helped me. So I watched all the different Hutchinson talks before giving this, and Phil Boyd had this great word cloud of all of his collaborators, so I did the same thing. And these are all the folks who are on my papers or were on grants with me. And no surprise to have Krista and Melissa come through very, very prominently, but also Marianne Moran, who has just been a joy to work with. So I will leave this up. Thank you for your time, and there's 1 minute if you have any questions. realize I stand between you and lunch, so feel free to do, oh, there's one question. Yeah, Danny.
Hi, Liz. Thanks a lot for that. The new methods, on the one hand, they appear to be increasing what you can observe chemically in the mass spectrometry, but on the other hand, they're also collecting more material on the resin. Sorry for saying this so slowly. To what degree is the benefit been to increase the proportion of DOM that you're holding onto for analysis versus the analytical improvements in detection at the mass spectrometry end of things? And I'm just going to back up. And at the same time, what prospect do you have? What proportion of DOM do you think you're currently collecting? And what are your prospects for increasing that proportion to make sure you're getting a representative picture? Thanks.
Yeah, thanks, Danny. Okay, let me see how I can unpack that a little bit. I will say that because the sample volumes are so much smaller with the new methods relative to the older method, we actually have a much lower background problem because 25 mils relative to 4 liters means that the background is almost negligible. So the smaller volume helps a great deal, cutting down background and increasing the molecules that we're interested in. In terms of how much we can collect, the reason I was careful to talk about this in terms of the biological DOC that we get in cultures is because the labile DOC in the field is so incredibly small. I will never be able to get a representative portion of field DOC because it is dominated by the refractory pools, right? And that's, at some level over the course of my career, that's the part I'm leaving behind, and there are great colleagues working in those areas who are going to make really important contributions in our understanding of the structure of that material. My focus has become much more on what is the material that's being generated by the microbes in the surface ocean, and I want to be able to collect those samples at small enough volumes and quickly enough that I can measure their turnover rates, because the flux of those materials should be about 80% of the DOC turnover in the surface ocean, but it's only like 5% of the standing concentration. So I'm trying to get at flux, and that's my goal. Thanks, Danny. Okay, there's a blinking red light, and it says 000. So I'm going to thank you all very much. Oh, I'm sorry, Claire, I didn't see you there. Did you want to ask one quick question?
I think this is a timely question. As you explore the ocean COC pool and these metabolites, are you seeing any products of degradation of plastics that you can identify? And I'm sure that's a whole nother research area, but.
Yeah, great. That would show up in our untargeted methods, which I haven't talked about, and we have not screened it systematically for plastic degradation products. So they're probably there, but we don't know about it yet. Okay. Thanks, everybody, very much, and enjoy your town halls, your lunch, et cetera.